some disturbation. I want to talk today about dark fermentation and bioreactors and hydrogen production strategies based on work of Professor Arto from Thailand and Professor Iqbal from Pakistan, Lahore, with cooperation with Polish scientists from Civil and Environmental Engineering of Gdańsk University of Technology, Professor Poczka. The work is based on his work is published in 2023 in International Journal of Hydrogen Energy, while the production of while the Professor Ardo work reviewing by hydrogen from the bioalgal cells through fundamental mechanisms and enzyme factors that engendering new challenging and prospects is from Fuel from 2023. The work released of Professor Iqbal is about the comparison of different hydrogen production by bacteria and mostly is microelectrolysis, dark fermentations, and photofermentations as three major. However, he chooses that as swing absorption and as about storage, about separation, chemical separation. But he didn't mention about membrane technologies that are already working. So this separation is somehow a trouble that work less because pressure spring absorption is method that is using much energy because separation of hydrogen was troublesome but also requiring much energy, and membranes which are being designed are better because they not require such energy. This is the dark fermentation, and only he mentioned about dark fermentation is advantage as 33% of hydrogen production efficiency due to Thaler rule in natural way. However, in Thaler rule is some exception in case of genetically modified by Professor Iqbal, but from Vienna Institute, not from Pakistani Lahore, because they are two different. And these work in case of Professor Iqbal from Lahore and Professor Boschke say about using as a biogas plant the bioreactors with packing, with batch reactor, but he doesn't mention any packing reactors. As in his feeds industrial waste sources chose agriculture, industrial, and municipal and forestry. However, industrial as Polish syrup and inhibitors is low pH. In all mentions, agriculture is mostly lignocellulose waste, which is especially at forestry is very hard to be pretreated. He mentioned about pretreatment of this waste. He chose that pretreatment of inoculum about gamma irradiation. But this is chosen is only in China, in some bacteria from Beijing, which were coming from from nuclear plant reactor and found that this method, radiation, is also useful besides pretreatment, but mostly is pretreatment in case of 550 to in case of dark fermentation, because he chose waste production as how to prepare biological layer only for dark fermentation, not to other methods like photofermentation. The problem is also that he mentioned about microbial electrolysis, Professor Boczkai, while in case of Professor um, Sir Ardo is using also as one point as microalgae, which doesn't mention because lipids how initially as dark fermentation feed in this waste. Uh, he mentioned also about algae later, but not so few well. She mentioned about for biophotolysis, that is production, but isn't she doesn't show that these algae also producing some hydrogen, and this biophotolysis and fermentation or photofermentation can be mixed together, like Professor Liberatos make and like Professor uh, Ardo make in his research, because he chosen also these three methods, microbial electrolysis, cell, and but he combined this method, not unlike, and microbial electrolysis cell is useful because Professor Boczkai he find only using microbial electrolysis cell as for splitting water, but he doesn't show that in work like in work of Professor Arthur, is microbial electrolysis need produce enough voltage to spill ammonia and some low organic acids, because it's true that water is too high electronic voltage. But in the case of several big variety, this case water needs only 0.3 and microbial and some organic acid, which are less of dark fermentation, even lower. Like use Professor Ardo for this combining of all methods, which are gas, which are in cultivating, they capture carbon dioxide. Then in the next stage, after using, they can use during this put some hydrogen of biophosphates, and the rest can be used as for lipids, as for bioethanol or biofuels, or biobutanol or lubricant. That is what work of Alto. Why Professor mm, Professor Iqbal from Lahore Institute is showing little about, only some little about biodiesel, not biofame and bioplastic. These two methods, he also mentioned that photofermentation as supported. It is, but it is using mostly all substrates like that fermentation, but it requires light, which is showing that's lowest stability than dark fermentation. And can photofermentation use these acids, which are used or not used or in lipids or in dark fermentation, to make all effluents optimized for hydrogen production, and then he shows that this hydrogen can be used as an digestion help. So these are differences which I use. He used about continuous standard steel reactor, an aerobic fluid reactor, and membrane reactors. All are useful, but not now are very developing some packing bed reactor and change a combination into flow, because their idea is to use some second combination of many hydrogen production to be made optimized. Also, as little mention about nanoparticles and and what that about sugars that are main source, but he doesn't show that lipids and also protein can be also used. So this is the case which is two worlds difference in the analysis. And this shows the good position, good uh, uh, good discussion, those words. Some more, some wider, but shows the expand and development of this branch of science. And thank you very much for listening to me.